Hello and welcome to the first video in the series of DNA and RNA. We are going to start off with the structure of DNA. Now, what DNA is, it stands for the word deoxyribonucleic acid. And we get the initials DNA from the D in deoxyribo, the N in nucleic, and the A in acid. What the structure of it looks like, what it is built as, is it is three different parts. It's built out of things called nucleotides. And a nucleotide is this right here. That is a single nucleotide. It is built out of a 5-carbon sugar, which we're just going to call a sugar, a phosphate group, and a single nitrogenous base, which we're just going to call a nitrogen base. A single nucleotide is all three of these built together. And DNA is a series of nucleotides attached to one another. So it looks similar to this. You have the backbone here of the DNA built out of the sugar and the phosphate. And you have two series of these back to back with the nucleotides touching, kind of like the rungs of a ladder. And the shape of DNA is called a double helix because while it is shaped like a ladder, it is also shaped like a twisted ladder, with the rungs of the ladder passing in between the backbone here. So while it is shaped like a ladder, you can look at it like this. It is also a twisted ladder in which you would take that and you would twist it around so that it is in a spiral, hence the helix name. And since there are two sides to this helix, it is a double helix as the shape. Now, with these nucleotides, you have the phosphate group and the sugar. That forms the backbone of the DNA molecule. You also have your nitrogen base. And there are 4 different nitrogen bases that you can have in DNA. The first one is adenine, which when we look at it in DNA, we will abbreviate by the first letter A. We then have guanine, which we will look at the first letter G, cytosine, C, and thymine, T. Now, these bases also match up in specific ways. Here you see the two bases joined together from each side. Depending on what one side is, determines what the other side is. They only match up in specific ways. For example, guanine will only match to cytosine. And adenine will only match to thymine. So if you have a series where you have adenine, guanine, cytosine, thymine, adenine, it will only match up if A is up here, the strand that it connects to will be thymine. G will only have C on the other side. This G will only have C on the other side. This C will only have G on the other side. T will only have A, and we are back to A with T. They only match up in those specific ways. So that is all that we need to know about the structure of DNA. The next video we will be doing, we'll be looking at how DNA copies itself. During cell division, the DNA has to copy itself so that each new cell can have a copy of the instructions of how to run it. So the next video we'll be looking at how exactly we achieve that.